Phased microscopes are going to be really good for examination of live specimens, and then also for studying and examining specimens that would be damaged by the use of stain or by attachment to slides. So ultimately, what the phased microscope is going to do is it's going to alter the phase or the sinking of the rays that are passing through the specimen and through the lenses. And this will be used to enhance the contrast of the sample. And since it increases the contrast of the sample, we can get away with not using the stains. So if we look at what we mean by phase or sinking of the rays, the first picture here shows rays that would be in sync with each other. So if I just mark the peaks of these, notice how the one on the top is at the same sinking as the one on the bottom. If we look at the other picture, this would be out of phase. Notice that the peak of one is off from the other one. So they are out of phase or out of sync with each other. If we have rays that are out of sync like that, then that is going to enhance the contrast that we get. So that's what the phase microscope is going to do. We have a special phase plate, which is going to be something that the light is actually going to pass through. When the light passes through that phase plate, it's going to alter the sinking so that they will be off from each other, similar to what we have up here, and that ultimately creates extra contrast so that we can better view those specimens. If we compare what a specimen would look like under bright field microscopy, and we compare that to dark field, and then finally to the phase contrast, you can see that the specimen in these pictures, it's magnified exactly the same amount with every one of them, but we're able to view the specimen a little bit different. We can see different characteristics. With the bright field image, the whole thing is very bright in appearance. Sometimes we cannot get the same kind of contrast that you could get if you look and you compare that to the phase contrast. And then with the dark field imagery, we have again the dark background, and we're seeing the specimen on top of that dark background. A fluorescent microscope is a step up from a light microscope. With a fluorescence microscope, instead of using visible light, we're using UV light to fluoresce objects. The benefit of this is that it is going to increase the resolution, and the reason why it's going to increase the resolution is because UV light has a shorter wavelength, which means it's higher energy wavelengths, and the higher the energy, the greater the resolving power that we have. So in this image here, we can see the viewing of a specimen using a fluorescence microscope. Sometimes when we use fluorescent microscopes, there is actually a use of antibodies that will attract specific fluorescent dyes. So fluorescent dyes often have to be used, and then when we combine the antibodies with the fluorescent dyes, we can specifically label or attach these to specific regions of a cell or specific regions of a molecule so that we can specifically identify regions that we want to study and be able to view in the microscope. So in this picture, you can see that these particular antibodies that are getting the dye attached to them, the fluorescent dye, they are going to attach to the surface of the bacteria, and then later we're able to view that entire bacterial cell. Confocal microscopes are going to use fluorescent dyes or fluorescent antibodies, like we just looked at, but they're going to combine that with UV lasers. So in this case, the images are obtained from a single plane of the specimen. When we have this, we're going to have basically a series of pictures of that particular specimen, and then it's a single plane, and then we would have to use some equipment to assemble those different planes together to give us a nice overall view of that particular specimen. Electron microscopes are obviously a step up from all of these. With electron microscopes, we can obtain fantastic magnification. We're looking at anything from 10,000 times magnification up to 100,000 times magnification. So significantly improved compared to our light microscopes. This is going to be because electrons traveling as waves have wavelengths that are extremely short. So they're way shorter than visible light, they are shorter than UV light, and again, the shorter the wavelength, the greater the resolving power that we will have. So electron microscopes will use the electrons. They will obtain a much greater resolving power and a much greater magnification, and these are used to view a cell's ultrastructure. What we mean by ultrastructure is studying the individual structures on the inside of a cell. Because we have such an enhanced magnification and resolving power, we will be able then to study ultrastructures of a cell. There are different types of electron microscopes. We have what are called transmission electron microscopes, and then later we'll have some scanning electron microscopes. Both of them are using electrons. In a transmission electron microscope, we're using a beam of electrons, and this is going to produce an image on a fluorescent screen. So if we just look at this right here, you can see that it is similar to what we're doing with the light microscope, except that we have an electron gun. So we're basically shooting the electrons through the specimen, similar to the way that we would be shining the light through the specimen. It passes through, we do have the refracting and the bending, and then finally at the end we're getting an enhanced magnification of our image. We look at this on a screen, and we can see really great structures, ultrastructures inside of our cells. Now, some things to know about the use of transmission electron microscopes. First of all, this requires the use of a vacuum. So we have to view these inside of a vacuum, which means that we can't put live specimens in there because they wouldn't survive the vacuum. The reason why we have to use a vacuum is because when we're talking about dust particles and things that we would have in the air, they can certainly interfere with the path of the electrons. Another thing is that the specimens have to be very thin. So we have to have special equipment that's able to produce these very thin sections of the specimen. Typically, we call it a microtome. A microtome is the tool that's going to be able to actually cut those very thin specimens for us. Again, you can't have a live specimen because not only are we placing it into a vacuum, but we also have to slice it up into pieces. And so a real limiting factor here, besides just the overall expense of getting the vacuum and using that, and then also the slicing, is that we are not able to view live specimens. So here's a look at a transmission electron microscope. So we have the microscope on one side, and then you can see that it is inside of a vacuum tube. It would be projecting the images onto a computer screen, and then you can see the image here of what we might be seeing with that transmission electron microscope. Really nice detailed structures. This is bacterial cells that we're looking at here, but the detail, the magnification, the resolution is so much improved compared to those light microscopes. The other type of electron microscope is the scanning electron microscope. This one is similar because it is going to use electrons, but this is going to rapidly focus the electrons back and forth across the specimen's surface. So that's the scanning part of it. And then this electrons are basically bouncing off of the specimen and they're being detected. So you can see that we do have a detector over here that is collecting those bounced electrons, if you will. This also requires the use of a vacuum. So we've talked about how that is going to be cost limiting, and it's also going to prohibit us from looking at live specimens. With this one, because we're depending on the electrons to actually bounce off, we have to coat the specimen with a special metal. So we have to coat the surface first so that the electrons will deflect off. And then whole specimens can be observed in this case. So that's an advantage here. These are not live specimens, again, because of the use of the vacuum, but we don't have to actually cut them into slices using that special microtome. So we can look at a whole specimen and get a view of that. One last type of microscope to introduce here is what we call probe microscopy. Probe microscopy is a relatively new advancement, and this is going to use tiny electronic probes that are able to magnify more than 100 million times. So that is really fantastic, unbelievable magnification that we can get with these. What these are going to do is they have a tiny little probe, which we're showing in this image here, and that probe is going to measure the flow of electrons to and from the probe and the specimen surface. This requires the specimen to be able to conduct some electric current. So you can see in the image here that we've got different electric charges and things inside of our specimen. But since this is able to magnify at such a tremendous amount, this is going to reveal details at an atomic level. So we can actually see molecules when we're talking about probe microscopy. Again, this is a pretty new advancement. This is not something that's going to be real readily available to the average scientist, the average researcher.